Okay guys, we have talked a bit about enzymes. We've talked a bit about how they are necessary to provide us with an ability to break bonds and to make bonds at a certain stable temperature. And now what I'd like to do is focus in on a sugar molecule. Let's use glucose as an example of a molecule that we want to break down. So we have C6H12O6. We got 24 atoms in there, carbon, hydrogen, and oxygen. We know those are the elements in carbohydrates. And we want to break those bonds to liberate that energy. And we want to harness that energy. We don't want it all to go away, all right? So before we get into that, how a living cell would do that, let's look at how we might do this in a chemistry laboratory, right? We can do certain things in a chemistry laboratory that could break bonds. And so let's look at a video here and then we'll get back to it afterwards, all right? Okay, so you can see there that the sugar in gummy bears has a lot of energy, right? And they have this potassium chlorate. This was a compound that was capable of breaking the bonds that were holding the sugar molecules together in that gummy bear. And the energy certainly was not harnessed, right? The energy was liberated in the form of light, heat. You could see what looked like smoke, and some of it probably was smoke coming out of that tube, but it was a lot of water vapor as well. There was water being released, carbon dioxide being released, and some carbon that formed inside the tube. That black material was carbon. All right, so when we eat a gummy bear, obviously that can't happen, is not happening inside of our digestive tract, right? We're still breaking down that gummy bear though. And that reaction that you saw in the tube there, that could have occurred with an M&M or any sugar-containing molecule. And they did warn that if you had any organic material at all in that tube, that the potassium chlorate would work upon that as well. So it's going to break bonds in all sorts of organic molecules. So what are we going to do in the living cell? When it comes to metabolism, we want to break the bonds in glucose and we want to harness that energy. So we want to break bonds, that would be catabolism. So what we want to do is take glucose and through a set of reactions where we would break bonds all the way down and release the energy from those bonds, okay? The energy from bonds that are broken in glucose can go into forming a bond between a molecule called ADP and a phosphate ion, okay? That would form ATP. Now the bond that is forming that molecule, ATP, is now holding on to the energy that was released when the bonds holding the carbons, hydrogens, and oxygens together in that glucose molecule. When those are broken, other bonds are made in this ATP molecule. So it's really just a matter of we need to break bonds to make bonds. All right? Now, on the flip side, what we can do is use that ATP molecule in a living cell and turn around and re-break that bond. Break that phosphate off again. Now, the energy can be given up to form bonds elsewhere. So, food we eat, sugars we take in, are going to be broken in a very systematic way, not all at once like you saw in that test tube, in a very methodical way using enzymes, and the energy will be directly put into bonds to form that ATP. In turn, later we can make bigger molecules using the energy from that ATP molecule when we break it. Okay? Now, we have terms that we use besides catabolism and anabolism here, right? In this case, we know that glucose is going through a catabolism process, and over here on the other side of the ATP molecule, it is also catabolized in red. Okay? What we can say is the arrows in red are exergonic. Energy is being released, right? And we can also say that these bonds have been oxidized. They have been broken. Okay? So oxidation means that we are breaking bonds. And then the blue arrows, okay, we say that these are endergonic reactions. Energy is being put in as bonds are forming. And this is a reduction part of the process. Now keep in mind, electrons are moving around here, right? When I break a bond, a covalent bond, there are a couple of electrons involved with that. And those electrons will go into making bonds in another place. Okay? So a lot of times what they'll do is they'll talk about oxidation as a loss of electrons. Okay? So we say oxidation is a loss of electrons, a loss from the glucose molecule being broken down. And then we'd say that the ADP molecule that has another phosphate tacked on, so a diphosphate molecule to a triphosphate molecule, that actually gained electrons to make that new bond. So this would be a reduction, which is a gain of electrons. So sometimes people say oil rig, right? We have these paired reactions. The oxidation and reduction reactions are paired up so that the oxidation can give up the electrons and the energy, and the reduction side will make a bond. So throughout this cellular respiration process we're going to talk about in a second, throughout this process of breaking down sugar, we're really just over and over and over again breaking bonds to make bonds. Okay? So when we break these bonds, we're going to harness a good amount of that energy, but most of it will be lost as heat. In fact, in the human body and an animal, we have a lot of energy being given off as heat, especially in our muscles, and this is what maintains a body temperature, right? We're always emanating heat off of our surface, but to maintain the heat within us, we have this constant inefficiency actually going on within our muscles. Our muscles are breaking down these molecules, and a lot of it is lost as heat, but some of it is harnessed in the form of ATP. Okay? So we call this carbohydrate catabolism, right? The carbohydrate in this case was sugar, right? So here we have a glucose molecule down here. All right, and we have those 24 atoms, C6H12O6, and they're being broken down. Now there are two ways to go here. That glucose molecule could be completely broken down. As we do so, we're going to harness some of that energy in the form of the ATP molecule. If we completely break that glucose down, we would define that as respiration. All right? No longer are we going to use the definition respiration like we did in anatomy and physiology, right? In A&P, you think respiration, you think about the lungs, right? About breathing, the respiratory system. Well, here, no system is necessary. If this is an E. coli molecule doing, or an E. coli cell doing this, then it could take that glucose, have the enzymes necessary to completely break down that glucose, and we'd say that it is performing respiration. Now, if that same E. coli only partially breaks down the glucose, only partially breaks down a sugar molecule, we would say that it's fermentation. Okay? So we could have glucose respiration, we could have fermentation of that glucose. Now, in the end, some of the electrons that are liberated in the process of respiration can be taken on by the oxygen molecule. If oxygen is involved at the end of the respiration process, we would say that that is aerobic respiration. Okay? If some other molecule other than oxygen is being used to bind those electrons that are released, we'd say it's anaerobic respiration. Okay? So this is to say that anaerobic respiration, that organisms can breathe, quote, right? They can perform respiration, but now we know it's not really breathing, it's about the breakdown of a glucose molecule. And they can perform this respiration in an anaerobic way. They don't need the oxygen molecule around to get this glucose molecule broken down completely. Okay? Now, we would be an example of an organism that is completely dependent on oxygen. We perform aerobic respiration, and we do not have the capacity for anaerobic respiration. All right? So what will we need to know about the basic steps of this process, whereby glucose, so here we have glucose, is broken down either completely or partially. Here on the right side of this image, we see glucose being broken down just part of the way to form fermentation end products. This is called fermentation, just a partial breakdown of the sugar. So a handy way of thinking about this might be a candy bar, right? You have a candy bar, this is your glucose molecule. Now you could eat that candy bar and completely demolish that candy bar, right? And break it completely down. That would be like respiration. But if you just kind of break it in half, you eat half of that candy bar, that's what fermentation is. You're not going to get as much energy out of it because you've only partially broken down that glucose or eaten that candy bar. Now, on the left side of this diagram, we see a set of processes that take us all the way through to the point where we see this oxygen molecule. This would be a respiration process, okay, where all of the bonds of that glucose molecule are going to be broken down. Now, what you need to know about the respiration process is listed on the side of this slide. Okay, you need to know 4 things about respiration in general. Right now, let's just imagine this is aerobic respiration, okay? This has to be aerobic because we see oxygen is involved in this process, okay? So let's say this is respiration taking place in one of our own cells, okay, in a human cell. I would want you to know the overall name, of course, which is aerobic respiration. But one of the things I'd want you to know beyond that would be the name of each phase of the respiration process. All right, so let's circle the names. So we'll keep it in red here. So the name of each phase. Glycolysis is where we get started breaking down glucose. Krebs cycle is the middle of the process where we're breaking down glucose. And the electron transport chain, or sometimes they say electron transport system, is the third phase. So 3 phases, you need to know the name of each of them. You should know the location that each of these stages takes place in. So if this is one of our own cells, like we're talking about, we want to know where in the cell does this take place. Glycolysis, this would occur in the cytoplasm, right in the cytoplasm of a eukaryotic cell. The Krebs cycle, this would take place in the mitochondria. And the electron transport chain or electron transport system would take place in the mitochondria. Okay, so location taken care of. If this was a bacterial cell, we know it wouldn't have mitochondria. So pretty much all 3 of those phases would have to take place in the cell, right? Partly in the cytoplasm, partly on the membranes of that cell, but definitely no mitochondria involved. All right, the third thing, let's go with an animal cell again, like our own, would be what are the oxygen requirements, right? So, question mark, is oxygen necessary for each of these 3 stages? The answer for glycolysis would be no. Oxygen is not necessary for glycolysis to occur. You can partially break down glucose, in other words, and not need oxygen for that to happen. But for Krebs cycle, or the electron transport chain, yes indeed, we need oxygen to drive this process of oxidation, to drive this process of breaking bonds, right? Oxidation and breaking bonds. We're oxidizing this glucose, and oxygen is definitely part of the process with aerobic respiration in these 2 parts of the respiration process. Finally, ATP production. What is the quantity of ATP molecules we get from each of these 3 stages? The answer for glycolysis, we only get 2 ATPs. Okay. The answer for Krebs cycle, again, we only get 2 ATPs. But then throughout the electron transport chain, we get in excess of 30 ATP molecules. 34 would be a maximum. Okay. So we're going to get a lot of ATP out of this process. If we get 34 out of the electron transport chain, add the other 2, we're getting up in the 38 range of ATP molecules, and that's our big payoff, right? The reason we're in this game is to get usable energy in the form of ATP, all right? So we need to know the 3 phases, what they're called. We need to know the location that they each take place in, the amount, the oxygen requirements for each, and the amount of ATP that each will produce. Now, I just want you to know, I'm not putting this slide up to scare anybody, but I just wanted to share this slide to show you that each one of these stages of respiration actually involves many enzymes where they're breaking down bonds, and it's not just one reaction, right? It's a whole host, a whole sequence of reactions taking place. So this is glycolysis, and we see glucose in the beginning here. And then it goes through the process here as we go around to ultimately produce a couple of much smaller molecules here. So you've basically broken the candy bar in half. But this is a more complex process than one enzyme. Okay, so I want you to know that it's a whole host of enzymes that an organism needs to possess to do this. Okay, the last thing I'd like to mention is how is it that this process would be different if we had anaerobic respiration going on? Okay? So, aerobic respiration, the process we just went over, would start with glucose. So here we have glucose, C6H12O6, right? 24 atoms held together. Oxygen is necessary, so here we see oxygen in the reactant category of this reaction. And we saw on the order of 36 to 38 ADP molecules turn into 36 to 38 ATP molecules. Something we can use later, right? We can break the bonds in those ATPs and make bigger molecules. Two of the end products of cellular respiration were CO2 and water. And of course, we get rid of CO2 by breathing it away, right? But a unicellular organism can also do this. The CO2 will go right through the membrane and that waste product would go away. But how is anaerobic respiration different, right? Here, aerobic respiration, we needed to have oxygen, right? That's the whole point. Aerobic. All right? And here we have an image where oxygen in the aerobic example, glucose went through glycolysis, Krebs cycle, electron transport system. Oxygen bound up the electrons that were liberated and formed water. Okay? 36 to 38 ATP is really lucrative, right? Lots of energy harnessed. Now, there are examples where oxygen is not the final electron acceptor. This is what we call oxygen in aerobic respiration. It is the final molecule that's taking on electrons that have been released through this oxidation process and makes water out of that. Okay? If a molecule uses some non-oxygen molecule, some other compound other than oxygen, to grab onto those electrons as a final electron acceptor, then we call it an anaerobic type of respiration. So some examples of molecules that do this are sulfur-containing molecules, nitrogen-containing molecules, carbonate. So these are inorganic molecules other than oxygen taking on those electrons. So let me give an example of some organisms that use other molecules other than oxygen to grab onto those electrons that are released as a result of respiration. Pseudomonas and Bacillus. Pseudomonas cause catheter infections. They make biofilms. They cause infections in the respiratory tract of people with cystic fibrosis. Pretty nasty organism. These organisms use nitrate. They will grab onto those electrons as a result of the sugar being broken down and form atmospheric nitrogen. So they'll just release nitrogen into the air as a result of their respiration process. Okay. Sulfur organisms like Disulfur vibrio. This organism uses sulfate as the final electron acceptor and forms hydrogen sulfide molecules. These organisms are pretty abundant in Yellowstone, and the hydrogen sulfide they give off smells pretty nasty, like rotten eggs. Okay, so that's evidence when you smell that there are some pretty busy and happy organisms that are using sulfate and giving electrons to that sulfate molecule as a result of their breakdown of sugars in the region. Finally, we have methanogens. Okay, methanogens like to break down their sugars and give their electrons to carbonate molecules and form methane. Okay, now these organisms can actually be used to release an energy-containing molecule then, right? Methane can be used to heat your house. And so these are a whole host of anaerobic respiration type organisms. And I want to just mention before we leave this topic that a lot of areas in our own bodies are not aerobic regions. We have plenty of regions within our own system that are anaerobic regions, where anaerobic bacteria can survive and do quite well. So just because we're an aerobic species and we do need oxygen for our cells to stay alive, there's plenty of places within the human system that are anaerobic places where bacteria such as these can survive. Okay? Okay, we'll leave it at that for now, and we'll get into a little bit about fermentation next.